Hello everyone. Hi, how are you doing? I hope everyone is doing good. So welcome to another video of plant growth and mineral nutrition. In this video, we will be talking about different types of growth. That is arithmetic, basically two major types, arithmetic and geometric growth we will be discussing. We will be understanding the growth pattern, the growth curve that takes place in plant. Okay, some basis of this we have already covered in the first previous video, and we will look at what is differentiation, dedifferentiation, and redifferentiation. That is how the young cells further go to form specific and differentiated cells of the plant, and how do they perform specific functions, what is the mechanism through which they become special cells. So there are three terms in plants. In plants, there can be dedifferentiation and redifferentiation also. We will see this interesting mechanism that occurs in plant as well. Plants, okay, plants, but plant cells. So let's begin. First is the types of growth. So as you see, two major types, arithmetic and geometric. So let's first understand what is arithmetic type of growth. Uh, Mitotic division takes place, okay. After mitotic division, only one cell continues to divide while the other differentiate and mature. So if you have a parent cell and this parent cell divides into two daughter cells. So only one cell, this undergoes again mitotic division, and this gives rise to two daughter cells again. And this goes to form, it further goes into differentiation pattern and then maturation and all that developmental pattern. It will not now, it will not undergo cell division. So out of the two, only one goes, and then again this tree. So this goes for differentiation pattern, and this will divide. So only one cell at a time will divide. Example is constant elongation of the root. Okay, so one cell and then the successor of that cell, the successor of that cell, they undergo division, okay, arithmetic growth, whereas the rest of the cells will further undergo development and maturation. So it's like half of the team, one team goes into team building of the plant material, and the other gives rise to more number of members, okay, like division. So arithmetic growth, if you see the graph, arithmetic growth is a straight line graph. The growth rate is constant. So there will be no too fast growth happening nor too slow growth happening. It will be a very constant and a same pace growth happening. Okay, so you will have a linear curve. So with respect to time, the number of number of cells will also increase. So as and when the time increases, the number also increases gradually. Okay. Okay. So this is the linear growth. When growth is plotted against time, a linear curve is obtained. But when we look at the geometric growth, if you look at the geometric growth pattern, what do you see? Initially, there is no growth as such. But after a certain period of time, you see a rapid increase. No? See a rapid increase. It's like uh, you know, slope that goes up. So in geometric growth, what happens is once the meiotic division takes place, both the daughter cells continue to divide and redivide repeatedly. The major difference between arithmetic growth and geometric growth is this. Here only one cell divides, continues to divide, whereas here this parent cell divided into two daughter cell. Both the daughter cell will again undergo cell division. Then this again will go cell division. So all of them are undergoing division and redivision on a repeated basis. So geometric growth. Initially, it will be slow. Initially, as you see, the curve is touching the line x-axis. Initially, it is slow, but later on, it picks up a very rapid pace, like the log phase, like the exponential phase that we studied in the previous video. A rapid growth will take place in this type of pattern. So this is known as geometric growth. What type of growth you see? A sigmoid curve. This type of graph which you see over here with respect to time, the growth shoots up after a particular time. This is known as sigmoid curve. Okay, when you plot growth against time. Okay, so this is what arithmetic and geometric growth is. If you look at this pattern, it is more understandable, this illustration over here. So in arithmetic, only one cell continues to grow, rest undergo maturation and differentiation. So as you see over here, this is how the pattern will look like. Okay. So this gray cell, the others go for maturation and elongation, and only one cell further undergoes division. Okay, this can be represented mathematically by an equation. You just need to memorize the equation. LT, that is length at a given time, will be equal to length at time 0 plus R into T. Okay, that is our growth rate. Growth rate, that is R per unit time. Okay, and here geometric growth, you see both the progeny cells resulted after the mitosis, they will continue to divide. So both the progeny, both the cells that are there, all of them will divide. So can you see the rapid number of daughter cells that you get when both the daughter cells are dividing? Okay, however, after a certain period of time, the nutrients will be limited. The growth will be, they will come to a stationary phase. Yeah, the growth will slow down, but from the initial period, the growth will take place at a very rapid. After a bit slow, it will pick up the pace. Mathematically, it can be represented by this equation. You can memorize this equation. W1, which is the final size, will be equal to, with respect to weight, height, and number, will be equal to initial size into e raised to RT. R stands for growth rate, T stands for time, E stands for the log. Okay, the base of natural logarithm. That is how you calculate your logs. So this is the geometric pattern of growth. The difference between the arithmetic and the geometric growth. Okay. So in plants, growth can happen via this arithmetic pattern also and geometric pattern also. Or some parts may show arithmetic type of growth, or some parts of the plant may show geometric type of growth. Okay, example we have mentioned over here, arithmetic constantly elongating root. Okay, where the elongation region of the root, there arithmetic type of growth happens. Geometric, here meristematic cells. Okay, meristems, wherever there are meristems, their geometric growth will happen, rapid multiplication and division of cells. This takes us to the growth curve. Okay, what are the different pattern different regions of a growth curve? Yes, so in the arithmetic and geometric, we saw in arithmetic we get a linear graph, in geometric we get a sigmoid curve. Okay, so what are this region, what is this different regions of this growth curve telling you about? Basically, growth curve is a study. It's a graphic representation when we want to study the total growth with respect to time. The total growth, that is the size or weight of the organ with respect to time or against time. Okay, so beginning with the zero time and zero number of cells. So this is the initial phase. We call it as the log phase. Growth is very slow. The cells are just budding. Okay. Though the growth is continuously increasing, but it will happen at a very slow rate. For example, the tip of the root and the tip of the shoot. The tip of the root, this is a root system, and this is your shoot system. Okay, so this is your shoot system. So the tip is growing slowly, and the of the stem as well as the tip of the root is also growing slowly. So in plants, the growth happens bidirectional. So the root against the gravity and root towards the shoot system towards the against the gravity, the root system towards the gravity. Okay, so example here is the growth of root apex and shoot apex. So slow and continuous growth is happening. It takes time. It is known as log phase. After that certain period of time, there will be a rapid growth. There will be a rapid growth. This phase is known as, sorry, this is lag, printing mistake, spelling mistake, L-A-G, lag. L-A-G, lag. And this is log or exponential. Another word is exponential. Very rapid, also known as grand phase by Sachs. Growth is constant. Example, fruiting regions of the plant. Fruiting regions of the plant can be taken as an example, where the growth is extremely rapid. After a certain period of time, that exponential growth will slowly slow down. So diminishing growth. Once, let's say, the fruits have come out, so they will take a now steady pace, sort of. So the growth gets a slowdown during this phase. And example, growth of plant after getting vegetative maturity. Once the vegetative maturity is reached, so now they will take a backseat, a little slow pace they will pick up. And once after that, after this phase, the plant or that particular region will reach a steady phase. A steady phase or a plateau phase, you can say. Growth will happen, but it will be very slow. Only maturation mostly takes place. Growth as such stops. The growth completely stops. Stationary phase. It is known as the steady phase now, wherein the tissue are now completely mature and they will perform their respective function. Okay, they will perform their respective function. So these are the different regions of a growth phase. Lag phase, okay, lag phase, log phase, and stationary phase. And in between the log and the stationary phase, you have a phase of diminishing growth. So which was earlier rapidly growing will take a slowdown, and once it reaches a bit of maturity, then those cells remain as it is. Now further growth doesn't take place in that particular region. Okay. This is all about the growth curve and the different regions of the growth curve. Now we move to the three terms that we need to understand. What is differentiation, dedifferentiation, and redifferentiation? Differentiation, time and again we are discussing that once the new cells are formed, to give them proper structure and function. As we discussed that some of the cells will go to become xylem cells, some of the cells will go to become phloem cells. So differentiation. Okay, then what happens to the what is then dedifferentiation? So okay, so these terminologies we will be seeing. Right now, I will not give you a gist of it. Some of the cells, once they are differentiated also, they can become meristematic cells also. So that's possible in plants. So let's begin with the first type. As you can see the picture over here, differentiation is talking about meristematic cells. Okay, they can become parenchyma cells. Then parenchyma cells can become sclerenchyma cells. They can also become parenchyma cells. Some of the cells can also become companion cells of the phloem tissue. Then some of the cells can become parenchyma cells of the epidermis. Some of the cells can become scleroid. Some can become tracheac. Some can become sieve tube. Okay, for example, simple example like meristematic cells can go and become root cells, can go and become leaf cells, can go and become the vascular xylem and phloem. So basically, the new cell is now having identity. The new cell is now having identity. It is having a purpose in life, like that. Okay. So a permanent change in the structure and function of a cell leading to maturation is differentiation. Okay, so obviously the structure of the meristematic cell, if it has to become a root cell, if the meristematic cell has to become a root cell, it will undergo change in structure, yes, change in function, and finally leading to maturation. and then now it will function as a root cell. This meristematic cell which has now become root cell, it will now function as a root cell throughout its life mostly. So it is maturation of the cells derived from apical meristem of root and shoot. So meristematic will be located at two places, at the root tip and the shoot tip. From there differentiation will take place. Okay, so during differentiation, the cells are undergoing major change in their structure, that is anatomy, major change in their metabolic activities, which changes both their cell wall and protoplasm. So in and out, not only outside but in and out, everything will be changed when a cell is a meristematic cell or a new cell is undergoing differentiation. Examples are here. For example, to form trachea elements, cells would lose their protoplasm, so they will not have their protoplasm from meristematic cells when they become trachea cells, okay, tracheal cells, which are part of the xylem. Then they also develop very strong elastic lignocellulosic secondary cell wall. So these are the features that the tracheary elements, the tracheary cells take. So the purpose of mentioning all these details over here is that from a meristematic cells which had different characteristics, different cell wall structure, different cytoplasm structure, now it has completely changed to very thick cell wall and having no loose protoplasm, okay, developing as a very strong elastic lignocellulosic cell wall. So the structure completely and drastically changes when a cell undergoes differentiation. One of the example that's mentioned over here. What happens in de-differentiation? Okay, sometimes the already differentiated cells, living differentiated cells, they lose their capacity to divide. And what happens is they go back to, they regain the capacity of division under certain condition. They go back to from where they came, like, let's say meristematic cells became parenchyma cells under differentiation. But under certain circumstances, they lost their capacity to perform as their parenchyma, or due to some external environmental factors, this parenchyma cell can de-differentiate again into meristematic cells. What happens in de-differentiation is basically the cells which were already differentiated, yes, the cells which were already differentiated. So let's say we were talking about meristematic cells, okay, they differentiated into parenchyma cells. So the meristematic cells, okay, they differentiated into, let's say, parenchyma cells or the sieve cells of the phloem. Okay, then due to some other, some certain conditions or due to some environmental factors, this parenchyma cells, for example, now lost the capacity to divide, okay, further, and that is they came to a stationary phase. In that course, now they regain, you can say regain, okay, regain the ability to become the meristematic cells again. And we know the function of meristematic cells. What is the function of meristematic cells? Capacity of division. Okay, so this process of, once the cell has meristematic cell has differentiated into one type of cell, but then sometime it can go back to become the previous version from where it came. That is basically it will become meristematic cells only again. It will not become something else. Okay, so this phenomena is known as de-differentiation. It is known as de-differentiation. One more example that we can take is mostly the interfascicular Cambium fork. Remember when we were talking about the uh, the inner layers of the cells, okay, where we had gone through various diagrams. So you have the epidermis layer, then you have the cortex, then you have the then you have the core, you have the cambium, and then in middle you have the pith, okay, and which conducts the xylem and the phloem. So this layer of cells, that is the cambium and coke, okay, they can be formed from the parenchymatous cell. Okay, so formation of intrafascicular cambium and coke cambium from parenchymatous cell present in the vascular bundles. Okay, so the living differentiated cells which lost the capacity to divide can regain the capacity of division again, and they can differentiate into some other types of cells. Okay, this phenomena is known as de-differentiation. Whereas what happens in redifferentiation, so we covered differentiation wherein the meristematic cells, they can become the root cell, they can become the sieve cells, they can become the leaf cells. And in de-differentiation, what is happening? Once the differentiated cell, living cells, can again come back and you know gain the capacity of division or some other function. Okay, so interfascicular cambium and coke, which are formed from parenchyma cell, is an example of de-differentiation. Another process that occurs in this context is redifferentiation. What is redifferentiation? The cells that are produced by dedifferentiation. So now we know through dedifferentiation, the cells are again, they regain the capacity to divide, for example. So once again lose the capacity to divide and mature to perform specific function is called redifferentiation. Okay, so now the interfascicular coke and cambium, let's say, it moved to form from the parenchymatous cell, they form the coke and cambium. Now that same coke and cambium can further, redifferentiate. Again, undergo the differentiation process. So, okay, so what has happened? See, from the meristematic, let's take the example. So from the meristematic cell, parenchyma cells were formed. So what happened? Differentiation happened. Okay, then this parenchyma cells now lost the capacity to further divide and give rise to different types of cells. So this parenchyma cells can now lead to formation of core and cambium, which have dividing capability. Okay, let's say the cells become pluripotent over here. So this, now they underwent de-differentiation. Earlier they were parenchyma cells, now they are something else. So de-differentiation. Okay, now again sometimes they might reverse back to form meristematic cells also, but here we are taking the example of parenchyma cells getting de-differentiated into coke and cambium, interfascicular coke and cambium. Now this coke and cambium can further re-differentiate. Okay, they came from already differentiated cells, now they can again re-differentiate, they regain the capability, they can re-differentiate and form something else. Okay, so let's say secondary xylem and secondary phloem, and secondary xylem, okay, uh, are formed from this coke and cambium. Okay, so secondary xylem and secondary phloem are formed from already de-differentiated cambium present in the vascular bundle. So you understand these three processes: differentiation, de-differentiation, and redifferentiation. Okay, so that's all with regards to these three terminologies. Okay, you can have a look at the layers of how the cells are arranged. So this we have discussed in earlier videos also. You can have a look at this diagram also. So you have this bark, you have this thicker layer. This forms your coat and cambium. Then you have the vascular cambium. Okay. The innermost is the pith, and this is your primary. The pink one is your xylem. Yes, the green one is here is the below the green line is your phloem. Yeah. So at the growing region, this is how the layers look like. As and when they mature, you can see over here as you move down, okay, and when you cut it, you can see the increase in diameter. So you have secondary tissues also, which will be formed from the earlier dedifferentiated cells. So that's all with this video. This is a summary of whatever we have discussed up till now. So let's just quickly go through the summary. Undifferentiated cells, for example, shoot and the root apex, okay, which are the meristematic cells. Now they get differentiated. They have they become specialized cells like parenchyma, collenchyma, sclerenchyma, primary xylem, phloem. This is what we have been talking time and again. When the meristematic cells, either of the stem, that is of the shoot, or either of the root, shoot and root apex, now they get differentiated. Once they are differentiated, they cannot have this capacity, okay, that is of the ability to divide into different types of cells. Then this differentiated cells can further de-differentiate. They are differentiated, they are already a type of cells, they are already a type of cells, okay, parenchyma, collenchyma. They can further undergo more specification, okay. So de-differentiation. And here this example, cambium, co-cambium, they regain the division ability, okay. Here they can divide and form different types of cells again, some of the different types of cells, okay. So now this de-differentiated cell have the division ability, which is regained. Then this de-differentiated cells can again undergo redifferentiation, like interfascicular cambium and co-cambium can now become different types of cells again, redifferentiate. Secondary xylem, so they can form phloem, they can form secondary cortex, but at this level now, they will lose the ability of division. Now, further after this, they cannot de-differentiate, and they do not have the division capability. The cells once made, they will not be able to divide and uh, increase the number of cells as such. Okay, so this is the summary of differentiation, de-differentiation, and redifferentiation. So that's all for this video. In the next video, we will be talking about development of the plants in general, plasticity, what is plasticity with respect to plants, and we will be talking about plant hormones. Okay, we will introduce ourselves to the plant hormones, and we will be talking about the first plant hormone, that is auxin. So thank you, take care, bye bye, keep revising. See you in the next video.